All right, class, as promised, we have a little video here to wrap up chapter 5. So when we return to class on Friday, we can start in on chapter 6. So the topic we need to talk about here is electricity and how that's going to relate to equilibrium in and around the cells. First, we have the law of conservation of electrical charges. The idea here is that for every positive charge, there's a negative. There's an equal number throughout. And while we can make ions with positive and negatives, those are all going to balance out in the end. Also, opposite charges attract. So positives like negatives. They do not like other positives. So there's going to be an attraction and a repulsion depending on what those particular charges are. If we need to keep positive charges away from negative charges, that's going to require energy. They're drawn to each other. There's that potential energy that creates movement. So keeping them apart is going to be difficult. And finally, conductors versus insulators. Conductor is just any substance that allows two opposite charges to move towards each other. So in our case, water is an excellent conductor. An insulator, on the other hand, is something that keeps those charges separate. So also in our example, in the human body, we have cell membranes, which do not allow ions to pass across from each other, so they keep those positive and negative charges separate. So here we want to look at an example cell. In this case, the electrical and chemical balances are in equilibrium. So as you can see, we have a neutral charge inside. The positives and negatives are balanced. Outside the cell, there is also a balance between positive and negatives. We also have a balance between our yellow positive ions and our blue negative ions with the outside. So there's no concentration gradient. There's also no electrical gradient. What we also see is that we have the cell membrane surrounding those 8 ions in the middle. So they are not allowed to pass across to the outside. So we've created a compartment specific to that cell. So now in this slide, we're going to change our example a bit. Here we have a pump that's going to allow one of our positive ions to be moved across the membrane out into the extracellular fluid. So if we look at this now, there is one more positive ion in the extracellular fluid, making it slightly positive. We'll give it a +1 charge. Inside the cell is now negative because it has one more negative ion that has positive ions. So we'll give that a -1 charge. So now we are no longer in chemical equilibrium. We also are no longer in electrical equilibrium. Now, this is basically what we see in the cells in our body, although it is a little more complicated. But one thing to keep in mind is that for our purposes, we're going to assume that the extracellular fluid is at a 0 charge. Now, that may not be particularly true, but we're looking at a relative scale. So we're interested in how different is intracellular fluid from the extracellular fluid. So if we use our previous example, instead of +1 outside and -1 inside, we're going to say outside is 0 and intercellular fluid has a -2 charge. Now, in the body, our cells look more like the example here. So in this artificial cell, it is full of potassium ions, giving a positive charge, and proteins which carry a negative charge. It's then immersed in a substance that is filled with sodium ions, giving a positive charge, and chloride ions, giving a negative charge. So this is fairly similar to what we'd see in the body, although it is simplified. The idea here, though, excuse me, is that to start, we still have electrical equilibrium, even though there is chemical disequilibrium. Now, as we've talked about, we have a variety of protein channels and protein carriers that allow ions in large molecules to cross the membrane. So if we add that membrane potassium leak channel, potassium is going to be allowed to flow out of the cell with its concentration gradient. So there's a lot of potassium inside the cell, there's none outside, so potassium is going to want to move out of the cell into the extracellular fluid. Now the issue here is that once the potassium goes outside, it gives a relatively positive charge to the extracellular fluid, a relatively negative charge to the intercellular fluid. So while the concentration gradient wants to continue moving potassium outside, there's an electrical gradient that wants to pull the potassium back inside. So we actually have a balancing of these two forces that doesn't allow either chemical or electrical equilibrium. Now in this next example, we've removed our potassium channel. So potassium is going to be stuck inside. Now, it's not going to be allowed to move. We've replaced it with a sodium leak channel. So through this sodium leak channel, sodium can move from the outside of the cell into the intercellular fluid based on its concentration gradient. Now, once we have that movement, sodium goes in, it's going to increase the electrical charge inside the cell, as well as its concentration inside the cell. Now, as you can see, it still has a concentration gradient pushing it into the cell. However, because the electrical charge is higher inside, that positive electrical charge is going to resist to the movement of sodium into the cell. So when our difference is 60 millivolts, so the cell is given a voltage of plus 60 millivolts, sodium is in electrochemical equilibrium, meaning that the electrical charge of the cell is exactly opposing the concentration gradient of sodium. So that while neither is in equilibrium, together they both balance out. Now here we have an example that it's a little more relevant to our own cells. So here we have our sodium-potassium pump, centrally located here, pumping sodium out of the cell and exchanging it with potassium, bringing it into the cell. We also have sodium leak channels, which basically allow sodium to sneak back into the cell, and potassium leak channels that let potassium leak out. We also find, as explained before, our extracellular fluid is given a value of 0 millivolts, and our cell itself is 70 millivolts less than extracellular fluid, so we'll give that a relative value of negative 70. So what we have here is this constant exchange where we're pumping potassium in as it flows out. We're also pumping sodium in as it's flowing in. And when we look at your questions at the bottom here, what force promotes sodium leak into the cell? Well, sodium is going to want to flow into the cell based on its own concentration gradient, also based on the electrical gradient. So the positive sodium ions want to flow in to balance out chemically, and they also want to go towards this negative 70 value. Potassium ions, on the other hand, they're going to be held into the cell by this negative 70. So the positive potassium ions are attracted to the negative charge inside the cell, but they're also going to want to flow out of the cell along their own concentration gradient. So when we get this negative 70 value, it's because we have this balance of sodium leaking in, potassium leaking out, and then sodium being pumped back out of the cell and potassium pumped back in via that sodium-potassium pump. And we can look at this graphically. This is going to be really important when we talk especially about nerves, is that we have this negative 70 value value is our resting membrane potential. That's what we'd expect to see at the cells. And this will change based on small changes in the sodium and potassium levels inside and outside of the cell. We can have depolarization, where the inside of the cell gets more positive, although the value at this point is still negative. If it moves back down towards our resting membrane potential, it's called a repolarization, so the cell becomes more negative. And in the case where the cell gets more negative than it's normally found at resting, we call this hyperpolarization. So this, excuse me, at a negative 80 millivolt value is more negative than we might normally expect to see. Now, this will be really important when we start talking about the nervous system. We'll talk more about this. But another thing to remember is we've talked about how some of these ion channels are voltage gated. So they can be closed or open depending on the particular channel, but a change in voltage can cause them to open or close. And that's what we'll see in a few instances. So here we're going to look at the beta cells in the pancreas, which are responsible for insulin secretion. And they're going to be responding to glucose in the bloodstream. So with low levels of glucose in the blood, metabolism slows down. There's less glucose outside, less gets moved into the cell by facilitated diffusion. Metabolism slows down as a result. With lower metabolism, we have lower ATP. And we have these potassium channels that are open. Potassium is allowed to leak out of the cell. And with that leakage, the cell remains at its resting membrane potential. So we have these voltage-gated calcium channels, which when they're open, allow calcium to move in and signal a message. Now in this case, there is no calcium, there is no message, and we see no insulin secretion. In the opposite circumstance, high levels of glucose in the blood, so if you've just eaten, glucose rises. We have an increase in glucose transported in the cell, increasing glycolysis, the citric acid cycle, overall increase in metabolism, and as a result, an increase in ATP. With that high level of ATP, it's going to close these potassium channels. So these potassium gated channels respond to ATP. They're a chemically gated channel. And because those potassium channels close, potassium is not allowed to flow out of the cell. Without that outflow of potassium, we can have a rise, an increase in the charge of the cell membrane, so it becomes more positive. And that'll depolarize the calcium channels in the membrane there. So the calcium channels are responding to an increase in the voltage as it becomes less negative. It opens, allows calcium to flow in, and calcium inside the cell is a very strong, very important signal. We'll talk about it more in chapter 6, but in this specific case here, it's going to signal those vesicles to release insulin into the bloodstream, which is an excellent response to high levels of glucose. It tells our adipose cells, it tells our muscle cells to insert more GLUT transporters to absorb, transport glucose from the blood into those cells for use or for storage. And that is imperative in order to maintain blood glucose levels. Now in the next chapter, we'll talk more about some more examples of how all of these parts interact, how we have channels, we have carriers, and communication and signaling between cells, as well as the signaling from outside inside the cell. But the important thing to remember here is that all of these factors are going to be working together. So we've talked a lot about these things independently, but they're all going to come together, and being able to put them together is going to be really, really important. None of them are necessarily all that important on their own, but together they make life possible. So with that, we come to the end of chapter 5. Keep in mind, there was a lot of stuff in here. I think at face value, a lot of it seems very simple, very straightforward. But I think you'll find that it's not necessarily all as easy as it seems. So keep that in mind. This will certainly be on the test on the 24th, and we'll start on chapter 6, and we'll see where we get through that. All right, well, thank you, and I'll see you on Friday.